An overview of the material: a sterile swab, a total of 8 razor blades, 8 sterile stick applicators, a yeast suspension for the inoculation of 4 Petri plates, and at the back we have a cleaner solution, beakers for proper disposal of blades, tweezers, and 70% ethanol solution. We need to label the Petri plates. Make sure to write the information on the bottom part of each plate, not the lid, and follow the guidelines that are provided in the worksheet to make sure you can easily retrieve your plates next week. And the 2 light treatments should be clearly indicated. One will be kept in darkness and the other one will be exposed to UVA. For the inoculation of the Petri plates, a sterile swab is needed. The same swab can be used for the inoculation of all 4 plates, and we'll also need yeast culture. Notice that only half of the cotton part of the swab is immersed into the yeast solution. That's plenty enough to inoculate 1 plate. And the idea is to cover the whole surface of a plate. So you gently sweep over the surface. When it's been done once, you rotate the plate by 90 degrees and you repeat the sweeping process a second time. You would need to dip the swab again into the yeast suspension before inoculating a second Petri plate. We're ready to transfer the plant organs. Notice that we need 2 Petri plates for each plant species. 1 set of organs will be kept in darkness and the other one will be exposed to UVA. But let's first sterilize the tweezers using the 70% ethanol solution. You have access to a total of 8 razor blades and 8 stick applicators. 1 blade and 1 applicator per plant organ. There are 4 organs per species and a total of 2 species. That means 8 blades and 8 applicators. For the purpose of this video, we use a leaf specimen. The worksheet mentions to cut 2 pieces of about 6 millimeter in width. 2 pieces are needed because 1 will be used for the dark treatment and the other one will be needed for the UVA treatment. We then use a sterile stick applicator to press onto the leaf pieces, and it's important to press enough to physically damage some cells so that some liquid can be released. You need to release some liquid to be able to measure some toxicity using the yeast bioassay. If you're too gentle, nothing will be released, nothing will be able to diffuse onto the agar plate, and there'll be no toxicity at all. It's now the time to transfer the leaf pieces over our agar plates. When transferring an organ over an agar surface, it's important to ensure a good contact between the two. So position your organ, then gently press all over its surface so that some of its liquid can go through the agar. This is essential so that growth inhibition can be effectively assessed and measured with this bioassay. And repeat the same transfer with the second leaf fragment. Before proceeding with another organ, first dispose of your razor blade properly and transfer your applicator stick into the biohazard waste.